Welcome to Dr. Balaga's God Knowledge Doc podcast, your quick dive into the latest medical breakthroughs to keep you ahead of the curve. Let's get started. Today is 6 June 2025. This video is in a Science magazine article published yesterday, 5th June 2025, titled Gastruloids Enable Modeling of the Earliest Stages of Human Cardiac and Hepatic Vascularization. This is from Joe Wu's group at Stanford. Gastruloids are structured 3D aggregates of human pluripotent stem cells that mimic early embryogenesis. They're like tiny lab-grown models of early human development. They enable the study of primitive streak formation, germ layer organization, and early organ-specific differentiation. And these are made from stem cells that can turn to heart or liver cells. This study was used to model in vitro cardiac and liver vascularization up to Carnegie stages 9 to 10, which is about 3 weeks post-fertilization. The clinical relevance was the real embryos are hard to study, so insights into congenital heart and liver defects and vascular patterning from human pluripotent stem cells. This is a safe and ethical alternative. The Carnegie study is a standardized system of 23 stages used to describe the development of human embryos from fertilization to approximately 8 weeks, that is about 60 days of gestation. These stages are based on morphological features rather than chronological age or size. This table is a brief overview. In this study, roughly the model development corresponds roughly to Carnegie stages 9 to 10, that's around 3 weeks. And they were compared to Carnegie stages 19 to 20, which is roughly 6.5 weeks. Human pluripotent stem cells were micro-patterned 2 to 6 millimeters and differentiated using a vascularizing cocktail of VEGF and other factors. Triple reporter lines that include cardiomyocytes, endothelial cells, and smooth muscle cells enable live lineage cell tracking. Transcriptomics, single-cell RNA-seq, high-res confocal and 3D microscopy validate structure and lineage fidelity. A mix of chemicals help to create blood vessels in these organoids. Cardiac and hepatic vascularized organoids, HVOs and CVOs, were generated in a scalable and reproducible fashion. What are the results? There was patterned vasculature in organoids. The condition 32 yielded maximal co-differentiation of cardiomyocytes, endothelial cells, and smooth muscle cells. VEGF and angiopoietin 1 and 2 were the key drivers. Organoids contain myocardial, endocardial, epicardial, and neuronal cell types. Single-cell RNA-seq showed overlap with human embryonic heart, Carnegie stages 19 to 20. They looked very similar to human embryos at 6 weeks. These mini organs include tiny blood vessels with real branches and tubes. The heart-like organoids even had beating heart cells. Some parts were not perfect, like they were missing vein cells, that is, there was underrepresentation of venous endothelial cells, and some divergence from native in vivo transcriptional profiles. But yet, this was a big step forward. Clinical research and implications. This model enables mechanistic studies of human vascular development without ethical embryo use. Notch and BMP signaling, that is the bone morphogenetic BMP signaling, were essential for vascular branching. BMP inhibition was more disruptive than Notch. Potential applications: regenerative medicine, disease modeling, and drug screening. Establishes gastroloids as a tractable model for early human cardiogenesis and hepatogenesis. This is a powerful tool for testing new medicines safely, could lead to better treatment for birth defects or heart disease, and this should help scientists understand how our organs and blood vessels start to form. If you enjoyed this episode, share it with your colleagues. When we change how we see disease, we change how we shape lives. Until next time, stay curious, stay compassionate, and keep seeking wisdom. Podcast brought to you by Master MedFacts at mastermedfacts.com.